people haven't shown up yet.
Okay.
Are you ready? Yes. Okay. All right. Unfortunately, there will be people coming in, but all right. It's a pleasure to have Dr. Anna Yershova from Duke University today. She'll be talking about some of the exciting work she's doing on protein structure at Duke. Dr. Yershova received a BS in mathematics from Kharkiv National University in Ukraine, and her MS in computer science from Iowa State, and her PhD from Urbana-Champaign. And her thesis was mostly in robotics and motion planning, and she's basically extended her horizons in applying a lot of the mathematical tools and techniques to protein structures, and in the computational structure lab at Duke University. It's a pleasure to have her here, and the floor is yours.
Hi, everyone. So I'm very excited to be here today, and thank you for the invitation. And I will be happy to present my current progress in structure determination lab at Duke University with respect to the scope of the entire structure determination lab at Duke University under Professor Bruce Donald lab. Sorry, under Professor Bruce Donald supervision. So first of all, I want to motivate the problem of protein structure determination. High-resolution structures are needed for. Well, I have a little bit of a problem with the screen here. I wonder if I could.
Okay.
If that's okay. I could try to.
I think it has to do with the
resolution.
Yeah.
Well, let me try to fix it, because otherwise I may not be able to show everything.
I forget what resolution. I think it's 768 by.
Okay.
Let me just
Sorry.
find out
Okay.
exactly.
768. 1024.
1024 by 768.
Okay.
I mean, also the
I- I'm not
Oh
sure.
yeah. This is an American PC. So.
Yeah. I'm not sure why I don't get the properties, though.
Do you have the right setting for resolution?
I may not, but the properties does not show off for some reason.
You go under screen maybe. No?
Control panel.
Control panel.
Oh, yeah, I can do that.
I can't see
Sounds-
very well.
For
Where is it?
appearance and things.
Yeah.
Okay. Resolution. Yeah.
Actually, I can't see anything there.
It seems it's locked. Yep. Display. Huh.
Maybe hit the function F7 key if it's a regular laptop.
Oh, that's up in
Or is it F5?
F5 is this one, but we already did it because it puts the other screen.
Oh, you are on 1024 768. It says
Oh, really?
no.
Well, right here. Well, this is the right setting, I think. But I don't know why the screen resolution is locked for some reason. I don't know why, but let me see if it's too bad. I can actually do just the screen, then it will show everything, just will be smaller. Let me see. I probably can use just a screen, like maybe 85%.
I think that's fine.
That's fine.
Yeah.
Okay. It's better because then all of the slides will be.
Right.
Sorry about this. So determining protein structure is an important problem because when you know high resolution structures, then it's easier to determine protein functions and also do protein redesign for in drug design. And I want to talk a little bit about different ways of understanding protein structure. You can go from very low level, from the sequence level of a protein, and then this is the sequence, and in the geometric way you can see the sequence of amino acids as shown on this picture, and then the backbone is formed by the core of the amino acids, and the side chains are formed by the residues in each amino acid. Also, this backbone has some flexibility. Different portions, peptide planes, can rotate with respect to each other, and it can be defined by the dihedral angles between the amino acids. So this is primary structure of the protein. Then if you go a little bit, if you zoom out a little bit, you can see secondary structure elements in proteins. So the black here is the backbone, and because of the short distance interactions between the atoms, sometimes the backbone conforms to some kind of structure. In this case, it's a helical structure, and you can show it, people represent it using a cartoon diagram like this. So you can see this is the helix. You can see the helical structure if you look from another side at it, and this is also helix. So
can you use the cursor to point? Yeah. Because you can use the mouse. I think you can use the mouse there or something.
Oh, the mouse like this?
Or your own cursor.
Yeah, I can.
Yeah, because otherwise, it for people who watch it on on TV,
they will
they're
not
gonna
see my. Okay, so this is secondary structure of the protein. There are alpha helices in secondary structure. There are also beta sheets. But if you zoom out a little bit further, you see longer, more distant interactions make the protein fold into 3D structure. So here you see ubiquitin protein, which is shown in cartoon view. You see one alpha helix here, and another short helix right here. You see 4 beta strands, and the yellow pictures are of side chains, which are attached to the entire backbone on the protein.
This is at a finer scale, right?
This is in a finer scale, yes. And also between the secondary structure elements, you see in gray the cartoon view of the loops in the protein. Secondary structure elements are usually the most rigid portions of the protein, and the loops are the most flexible. So the goal of our lab is actually to discover the geometrical structure, the 3D fold of a protein, given whatever information we can obtain for a protein. And of course, we usually start with the sequence of the protein, and you can see that there is a huge gap between sequences that were discovered in protein genomics and the corresponding 3D folds of the proteins. So in the recent years, there are much more sequences discovered and very few structures deposited into the PDB, Protein Data Bank. Therefore, there is a huge motivation for discovering structures of the proteins, but also big motivation for high throughput structure determination of proteins. And our lab is working, this is the main theme of one of the main themes in our lab. So what are the current approaches for this problem? There are 2 main approaches. X-ray crystallography is one, and NMR spectroscopy is another approach. Both of these NMR techniques take a lot of time to collect the data about for each protein. Both of them require long expressing and purifying of the proteins. However, we strongly believe that there is much more future behind nuclear magnetic resonance spectroscopy techniques rather than X-ray crystallography for large proteins especially, and membrane proteins. the difficulty in X-ray crystallography is that growing good crystals is kind of black art, and it is hard to predict when and how a protein can be crystallized. However, NMR spectroscopy also has difficulties such as lengthy and very expensive time at the spectroscoper that one has to spend in order to collect NMR data. And also there are computational challenges in analyzing the obtained data. This is part of our lab. Bruce Donald is the PI. We have collaborator Peizo in biophysics. He helps with the spectrometer and collection of the data. We have currently 2 graduate students who are biochemists. They actually do the experiments in the wet lab, purifying and expressing proteins. And we have 3 students in computational part of the lab. And this is only an NMR project. We have more students and postdocs in drug design project of the lab as well. But this is the NMR structure determination part of the lab. So now, once I motivated why protein structure determination is important and why we are interested in NMR spectroscopy, I want to explain in more details what kind of data can we obtain for each protein. So there are 3 types of, 3 main types of data that people usually obtain in NMR. Chemical shifts. They are shown as green numbers. Oh, sorry, I'll show here. As green numbers near each atom. They usually, the values, the resonance frequencies for each atom, which represent kind of the ID of each atom in the protein. Then there is NOE data, which defines the distance between 2 protons in a protein. So for example, NOE would return the distance between these 2 hydrogen atoms, and also all other atoms, all other protons that are in close proximity to each other. And there is also a residual dipolar coupling data, which returns the global orientation of the bonds in the protein. So for example, if you have an H bond right here, RDC would return the orientation of that bond with respect to the global frame of the protein. And I want to go a little bit through each of the data in more details. So the chemical shifts, once they are obtained, they look like this diagram of resonances. And the problem is resonance assignment then. How do you detect which atom in the sequence of the protein actually represents each peak in the in this resonance diagram? Therefore, assigning chemical shifts is a problem, and it's usually done manually for now in NMR facilities. So biophysicists, they're able to do it manually, and there is no good automated technique for doing resonance assignments. Then the NIE assignment problem is the next problem. Once you know that, once you get the data from the spectroscoper, all of these peaks represent the 2 atoms which are defined on each of the axes by their IDs, by their resonances. So each peak represents 2 atoms being closed in proximity to each other. And the biggest problem, a very famous bottleneck in NMR spectroscopy, is assigning these peaks to particular protons. It's a difficult problem, and it's not really clear how to solve it. And I'll go into details. Why is it a difficult problem a little bit further? So the traditional structure determination from NOE would have the following protocol. first, resonances are assigned using manually. So resonances are assigned for each of the atoms in the sequence. Next, from NOE data, NOE assignments have to be done. And usually, right now, it's done either using molecular dynamics simulation, that is, from the sequence, molecular dynamics simulates how the protein would fold, and then the distances are estimated and compared against the experimental data. And then, using this technique, some kind of assignment is done, and then manual intervention is needed to evaluate whether that fold and that NOE assignment is good or not. So it's not really an automated process as well. And there is always some assignment ambiguity left after this process is done. But once it's done, you have a table, n by n square table, of all of the distances that are known to be present between protons in the protein. And from this distance, from this table, one can extract the geometry of the protein, the 3D fold. However, this problem of determining this geometry from the assigned distance table is known to be NP-hard. Therefore, the entire protocol is kind of doomed for large proteins, because the problem is NP-hard. Obtaining even the complete table, which would require n-squared entries in it, is impossible experimentally. And even if it's possible, then it always contains ambiguity. So there are many problems. And yes.
So that table contains all of the distances between the different components.
Yeah.
How do you estimate the direction? You have the distance, but it's in a 3D space, so how do you get
you don't have that.
You don't have the
You don't have that. Yeah, so it is hard. So it's like a 3D graph, and from the 2D kind of information about distances between the nodes in the graph, you have to reconstruct the structure. It is a difficult problem. Yes.
How big is N in a typical, in the picture that you got there? How many protons?
Oh, how many protons? Well, I think like normal proteins would have on the scale of 100 or so entries in the table, but some...
By proton, do you mean any nucleus? Or you're talking about hydrogen atoms?
Hydrogen. For
Okay.
Na is hydrogen. But sometimes, I mean, usually this table contains only linear number of entries in size of the protein. Therefore, it's never possible to obtain n-square entries in this table for large proteins.
And you have no angular constraints?
No angular constraint at this point. This is standard procedure from NAEs. And that's where, I'm going with all of this, is that many computational labs wanted to sidetrack this entire problem, this entire protocol of structure determination by, I will just repeat myself that from resonance assignments and NOE, using simulated annealing and molecular dynamics techniques, the initial fold is obtained, but there's like error propagation, local minima, manual intervention problems, which all result in ambiguous NOE assignments. And the structure determination from the NOE table is an NP-hard problem. Therefore, the biggest question, the biggest question is, can we really obtain a polynomial time algorithm with some provable guarantees on it, which would result in faster structure determination? And so how can we just sidetrack the entire problem? And the answer is using RDCs, the other type of NMR data, which became more popular only recently because of this new way of thinking about structure determination. And I'll talk about RDCs right now in more details. What is this data and why does it allow us to compute structures more reliably and faster? Yeah.
So given that you wanted to identify all parts of the 3D structure, are there ways to basically simplify the problem where there are certain parts of the protein that are more important than others? For example,
Yeah.
things that are exposed, so thus predicting more the binding sites versus any of the internal structure, and could that help in simplifying the problem any?
Yes, but you don't know where the binding site is even going to be. If you have just the sequence, that's what in this protocols we have only sequence in the beginning and some data with no geometric information whatsoever.
Do you think that at some point it would ever be possible to predict binding site based on just amino acid sequence?
Well, that's what they're trying to do using molecular dynamics, yeah. But however, it's not always reliable because proteins can fold differently, and depending on temperature, solution, and therefore once you put the purified protein solution in the spectrometer and you obtain the data, you really want to know what that conformation is. And that's why it's better to guide structure determination based on the NMR data that we obtain. However, what you're talking about is more structure prediction, and that's what you have to do if you don't have any other data. We do have the data, and we want to know how the protein is fold, what's the fold in this particular solution. So how do we do that? Of course, we have to base it on our data then more. Okay, so the RDC data. There is a little bit of physics, well, not a little bit, a lot of physics behind this, but I'll just try to explain it a little bit like in a simple way. So imagine that you have a magnetic field in which the protein lives. And you have a bond vector V, which represents some bond in a protein, and this protein is placed in the spectrometer in some magnetic field. Once you place the protein in the magnetic field, it starts tumbling. And each vector will tumble rapidly around this, each bond will tumble rapidly around the vector represented by the magnetic field. The average angle between the bond and the magnetic field vector will average to 0. Unless you place your protein solution in some alignment media. And that's when the RDC data can be collected. Once you place your protein in specific chemical medium, the ensemble average of all of the angles between the bond vector and the magnetic field vector will not be 0 anymore. And you can record some value, which is the RDC value D in this example. So the RDC value is some constants times the ensemble average of this expression, which relates the angle between the bond and the magnetic field. Therefore, each RDC value relates to the global orientation of each bond vector on which it is measured. Through some algebra, this expression can be actually, this is the algebra, it can be represented as a quadratic form of the bond vector and the alignment SOAP matrix, which corresponds to alignment medium in which this RDC measurement is collected. So this is the quadratic form, and V is the bond vector, which is a unit vector. S is a traceless and symmetric matrix. It contains 5 variables, 5 degrees of freedom. And therefore, since it's a quadratic form, you can think of this is where the strong geometric interpretation of the data, of the RDC data, can be represented. Vector V is a unit vector, so it has to be on a sphere, but it also has to be on the hyperboloid represented by this quadratic form, by the SOAP matrix. Therefore, each vector, if each bond vector, as soon as you collect RDC value, it has to be on the intersection of the hyperboloid and the sphere. And I drew this intersection as ellipses, but they are not really ellipses. They are spherical quarks. And therefore, once people realize that RDCs have really strong geometrical connection for protein structure, the new protocol was designed for structure determination. And RDC Panda is the software that was just released in our lab this January. And it uses the following protocol. It uses RDCs to compute with provable guarantees the global fold of the protein. So no manual intervention is needed at all. However, the limitations of this method are that the RDCs allow only to compute secondary structure elements reliably. You still need constant number of NAEs, which is much better than this n square number of NAEs, or even linear number of NAEs, which can usually be obtained in NA experiments. Just constant number, 3 or 4 NA values, allow to pack the secondary structures and then compute the loops in the protein. Just from RDCs and constant number of NAEs, global fold can be computed reliably. And once that is done, NAEs can be assigned the bottleneck in NMR spectroscopy. They can be assigned also reliably and in an automated fashion. And also sidechain placements. Our software also uses sidechain placements, smart algorithm to do that. So in the end, we have NA assignments in 3D structures computed in an automated fashion. So this is just to kind of highlight the advantages of using RDCs for structure determination. From RDCs, you can compute initial fold using exact solutions to the RDC equations that to those quadratic form equations that I showed before. And this allows avoiding the NP-hard problem for structure determination from NOEs. With the possible extensions to structure determination from sparse data, where you don't have enough NOEs to compute structure at all, which happens in high throughput settings and also for large proteins and other proteins where only sparse data can be obtained. And also it allows resolving NAE assignment ambiguity. And something that I'm not going to talk in this today much, but it also allows automated sidechain resonance assignment. So now I want to talk, I think I already mentioned that the current limitations of RDC Panda is that it requires very few RDC data, only 2 RDCs per residue in the protein, but it only can compute secondary structure elements, and NAEs are still needed to determine structures of loops. And it cannot handle missing values very well. So that's why I come to my project, to description of my current project. I'm working on, the overall theme is the same, improve current protein structure determination techniques in our lab, but also design new algorithms for proteins backbone structure determination using RDCs, so that no NAEs should be used at all. And this is the literature overview. I think I already covered different approaches. There are distance geometry-based approaches on NAEs and heuristic-based structure determination-based NAEs. But also many groups are concentrated on RDC-based structure determination these days because of the advantages that it offers. Now I want to talk a little bit more about the math behind RDC equations and how exactly it's used to determine structure of proteins exactly. So this is the quadratic form equation that I showed before. D is the RDC value, D max is the constant, V is the bond vector, S is the SOAP matrix. This is expanded quadratic form. And the interesting observation behind this equation is that if you know SOAP matrix, that is, you know everything about the alignment medium for the protein. So if you know S, V should lie, as I already described, the bond vector lies on the intersection of the sphere with the hyperboloid. However, if you do not know S, but you know the vector, fixed bond vector defines linear hyperplane in terms of the SOAP matrix elements. So there is this interesting structure to this equation. It's linear in SOAP matrix elements and quadratic in the bond vector elements. And this is something that we are going to exploit in order to resolve structure determination, resolve structures of proteins from this equation. And I just want to remind you that once you have the RDC equation, you just have the left term. Everything in the right term except for the constant is unknown because you do not know the coordinates of the bond vectors and you do not know the SOAP matrix. So therefore, just one RDC equation, of course, is not enough. And because one RDC equation has 7 variables. So you have to collect more data and so that you obtain a system of RDC equations which would allow solving it. So that you want to match the number of equations and number of variables. So now, since we need to collect several RDC values for the protein, let's see how this would be represented in the protein backbone. So this is the protein backbone again. If we collect many RDC values, then for each new equation, we have 2 more unknowns, because each new bond vector would again define an unknown 2 values. If the measurements are done in the same alignment medium, then S stays the same. So still, even if you have many RDC values, the number of equations is less than the number of variables, it seems like. However, we can use forward kinematics in the protein, which allows expressing each new bond vector only through 1 dihedral angles with respect to the previous bond vector. So in this way, we can reduce the number of variables in the system. So we can say that each consecutive bond vector can be represented through the previous one, either through 1 dihedral angle, or if there are no dihedral angles in between, it can be just expressed through the previous one. So there is this recursive structure to these equations. So again, this is the system of equations, but it can be written in this kind of hairy, it seems more messy way. However, this representation has fewer variables. It has only SOAP matrix with 5 unknown values, and then each new equation has 1 more dihedral angle as an unknown. So this already gives some hope, right? Because the number of equations matches the number of variables, except for that SOAP matrix, which introduces 5 additional unknowns that is not clear how to estimate. So there are different possible scenarios for this. First of all, some modeling can be used to avoid some of the issues arising from too many unknowns. Secondly, sampling for alignment tensors can also be used, but that sampling represents some kind of problem because then the automation is more difficult once you use some kind of sampling scheme in the algorithm. However, what is often done in current implementation of RDC Panda, we use 2 RDCs per residue to obtain secondary structure elements. However, we also need NOE, so we use some additional data to overcome this difficulty of having more variables in the system of equation than the number of equations. And I'm not going to go through the details of how it's done in RDC Panda. I just want to say that this approach was very successful, and the solution structure for the FF domain of human transcription elongation factor was recently deposited in the protein data bank. which shows that the method was applied successfully to determine structure of a novel protein. However, in my current project, what we do, we require, again, 2 RDC measurements per residue, but in 2 alignment media. Therefore, the number of equations is doubled, and all of the new equations contain the same equations as before with the same dihedral angles, but new SOAP matrix. This results in overconstrained system. Now we have more equations than variables, and as soon as the 9 consecutive bond vectors are measured using RDC in 2 media, then we have this kind of system, and we can apply the approach that we have developed. So now, how do we solve the overconstrained system? That is the problem, and that's what I've been working on. We have developed an algorithm which was partially implemented. We have partial experimental results for this algorithm. So the way we handle the overconstrained system is in the following way. Solving each equation in each term would be equivalent to minimizing the square of the square term, the function obtained from squaring the difference of the right and left values in each term, because, well, that's just natural. Therefore, and this is just expanded representation of the squared term for each equation, and it's been shown for overconstrained linear systems that solving an overconstrained linear system of equations is equivalent to minimizing the least square function of the terms. And this method is known as SVD. Therefore, what we do for our overconstrained system of nonlinear equations is just forming a least square function, which is an RDC fit function essentially, and trying to minimize it. And we minimize it using techniques which were developed also in RDC Panda, and this involves a little bit of algebra, but we found a nice way of finding local minimas in this function. So unfortunately, it's not a linear system of equations anymore, so we don't have just a hyperplane in the RDC values space. It's a more complex set of hyperplanes defined quadratically through dihedral angles. However, I'll not show you how we minimize this function. They result in a least square function. So, well, first of all, I want just to outline that once we can solve this minimization problem, then the advantages of this would be important in NMR, because we can then compute and pack secondary structure elements and loops without additional NOE data. We do not have to sample for SOAP matrix elements, which improves the which makes this method more automated without parameter tuning, and we can handle missing values in a more robust way. So this is the algorithm for minimization of that function. We have to start with current requirement is that the protein has a helix. Helical structure is very rigid, and the dihedral values of the inside the helices are usually bound to very narrow regions. Therefore, we could initialize our system of equations with the initial values with the average values for dihedrals in a helix. That gives a very, very good estimation for dihedrals. Once it's done, we can use, since the system of equation is linear in the SOAP matrix elements, we can use SVD to compute initial SOAP matrices. And once that is done, I'll show you how we compute dihedrals further in the iterative loop, which is converging using tree search and minimization. And I'll talk about this in more details later. But this is the initialization step here is important for the entire procedure because we have to initialize the system with something. And for helical structure, this is a very good approximation. Then for any other protein portion, we can start with the initialized SOAP matrices computed from a helix, and then complete the same procedure, which is an iterative procedure and convergent procedure. And now I will talk about how we compute the dihedral angles in each of the steps of our algorithm.
That's a good question.
Yeah.
For the minimization procedure, are you doing this for a particular reason, for example, speed or minimal complexity, and why didn't you use, say, any other type of global minimization algorithm?
Well, there are no global minimization is a very difficult problem, and this system has many equations. It has 18 equations, I think. Well, so for 9 residues, for 9 bond vectors, we have 2 RDC values. So it's 18 equations, and the problem is that it's linear in the SOAP matrix elements. However, these dihedral angles, it's quartic in the tangent of the dihedral angle, and they accumulate through the forward kinematics in each equation. So it's a really bad system of equations. I tried to write it down like in Mathematica, and I didn't have enough space on one screen, or even like, I think for the entire system, I didn't have patience to look through the entire system, through just the coefficients of the system. So it's really big. And there are no global optimization techniques known, except for one that I recently discovered and looked at, SOS polynomials. They seem to be able, and these SOS polynomials are sum of squares polynomials technique, which allows approximation of the system of polynomial equations with SOS polynomials for finding global minimum. That would be wonderful, but they are still not applicable for such large systems of equations. So there are no global minimization techniques known for polynomial equations like this that we know of. Therefore, we need to find other ways. And you know, in our lab, we really understand these equations well enough to have the following guarantee that I just wanted to present. That this is a very important point. Let me go back to the system of equations. If the data was perfect, then solving this system simultaneously would be possible because it wouldn't be overconstrained. There would be degeneracy, and but we know how to solve this system of equations when it's not overconstrained, because you can start iteratively. Each new equation has only one more variable. Therefore, you can solve equation for the first variable and continue in a tree structure, which I'll show here. Continue in a tree structure. So you start with first dihedral angle, solve the equation for it first. Then you obtain several solutions, and then you march down further through the system of equations. Since in each new equation there will be only one new variable, you can continue the process until you get the solution of the tree of solutions for the system of equations. However, this is how it's done when the system is not overconstrained. In my case, since it's overconstrained, we cannot just solve each equation because we have additional equations. It's like solving each equation would be finding an intersection of 2 ellipses. However, we have a third ellipse which corresponds to the new alignment medium that we obtained measurements in. Therefore, instead, we minimize the lists, this squared term, which is also a function of one variable only, and we can do it in each iteration of the tree, of the tree construction. Therefore, the argument is that if the system was perfect and the data did not have any errors, we would actually, in this way, find all of the solutions, because the minimization would be equivalent to finding solutions to the equations. However, we do not have perfect data. So there will be no, these three ellipses will not intersect in one point. We will have to use minimization, but the estimated structures in the tree will be close enough to the intersections of this function with the zero if the data was perfect. Therefore, we have really good starting. We have very good starting points for further local minimization. So that's the argument that we are using in our approach. That we just in order to solve global minimization problem, very often people sample the variable space, but it's very hard to estimate where to sample. Through this systematic search of the solutions of univariate functions, we have really good approximation of how to sample the variable space close to the minima of the function. So these are kind of the guarantees and advantages of this method. And I want to show you the preliminary experimental results on the helix and beta strand of ubiquitin protein. So this is the alpha helix. We had NH RDCs and CH RDCs collected for the residues from 25 to 31 in the helix. And we have applied this algorithm, and we have obtained the structure which is only 0.58 angstrom different from the X-ray structure deposited in PDB. Therefore, this is very good result because we didn't use any modeling. We just have implemented the minimization procedure that I have just described to you. And these are the back-computed RDCs versus experimental RDCs. They are very similar. However, for helix, it's much simpler because we have very nice initial approximation for the dihedral angles. For beta strand, we have, so far, I ran this experiment without using the further minimization along the tree. This is just the results of just one tree search, and that's it, without iteration. So this kind of shows what are the initial approximations for further local minimization, and it's pretty good because one of the branches in the tree resulted in this strand, which is only 1/15 angstroms far away from the X-ray deposited structure. And and this is the back-computed versus experimental RDCs. So it's a very good approximation. Starting with this, even if you run standard protocols such as NH Explorer or any local minimization technique, we could get to the nice strand. So these are good preliminary results, but this project is ongoing. So we hope to get more results for the entire proteins soon. And so in conclusion, I want to say that complete an exhaustive search over the space of all structures which minimize their RDC fit function seems possible now using this minimization approach, and is very promising for further development of RDC-based structure determination methods. And there are, of course, more extensions to this project overall, with the addition of other types of RDC NMR data, and further modeling such as dynamics of proteins and so forth. And we want to thank NH funding for supporting us through this project. Thank you.
Where do you run these algorithms? Do you use supercomputers, or does this run on your laptop?
This runs on my laptop.
A few seconds, minutes?
Yeah, because the tree search is really fast. It's only the number of iterations, which I don't know, because this is the part that is not implemented yet, the further minimization along those branches. So far, yeah, it's been just seconds. And similarly for RDC Panda, which was for a smaller number of RDC data.
So you wouldn't classify this as a seriously computationally intensive space?
No, even though it was for if we were to use NOE. Yeah. So this approach really has advantages in computational part as well as experimental, because you don't have to grow crystals.
Do you have to? There was some. I think it might have been in the other path where you showed molecular dynamic simulation was needed initially.
Yeah.
Do you rely on that? Or
We don't use it at all. Yeah, that's for NOEs, because you had to start somewhere. NOEs are not even assigned there, so molecular dynamics is used to see how it will probably fold, but then it's all verified against data, and that's where manual intervention is needed, because biochemists can look at it and say, oh, this is wrong folding, let's run MD again, and then they, So those are not really automated methods, and here it's highly automated. You just run it and you get the solution. We only fear of the following: if you get the solution with our method and it returns very nice RMSD function that is fit to the experimental data, but the structure is wrong. But we never run into that, into this kind of.
So that brings up the issue. In general, how are these things validated? Is this through the X-ray?
By biochemists. Yeah.
So actually, that was my question. How do you validate all this stuff?
Biochemists. Well, what we do, there are many structures which are known, so you can run your algorithm to see whether it will be reliable, and that's what we did. Against something known, yeah. And then what our lab did, they obtained new data for new protein, and they ran the algorithm. The algorithm returned very good fit to the experimental data. So how do you know? We deposited it because it seems biologically valid. There are no steric clashes. Like, you can also evaluate the protein structure using standard biological tools like mol. Like, there are a lot of software programs which can tell you whether there are steric clashes or whether the energies are good. Like, so yeah, you run against that as well, and then there is nothing else you can do.
So, so for this particular part right here, you ran it against a sequence that you knew was a helical structure. Or
yes. So I have a sequence. I have for each atom, for several bonds in the sequence, I know I have RDC values. And then I have a model of the backbone. That's the only input to the program.
And then in the next one, it was like a beta sheet or beta strand.
Yeah, beta strand only. Yeah.
Have you applied the algorithm to a longer sequence that, for example, has chunks of alpha helices and chunks of beta strands?
Oh, yes.
And then being able to, because now the initial starting point, the first one you chose an initial starting point that was really related to alpha helices.
Yeah.
In this one, it was a little harder to find the initial starting point. But what happens if you have these combinations, choosing the initial starting point and running through the algorithm? Have you looked at that?
That part of the project is not implemented yet, but what we are going to do is we actually are going to divide the protein into this set of protein portions, which we will apply algorithm to, and then we will use forward kinematics to connect them. So that's how we're going to do that. And for sure, if there will be missing values, for sure protein portions where not 4.5 residues are given with RDC data, we can already use the estimated alignment tensor for that. So there will be, again, the number of equations and number of variables will be good for solving the system. So that's the strategy, but we haven't implemented that yet. But that's how it was implemented in RDC Panda, where no minimization was used, but just exact solutions.
Any other questions? All right, let's thank our speaker again.